Hello, everyone. My name is Gerard Gatch, and I'll be the moderator and a presenter for today's webinar on the topic of analytical to prep scale tools for the purification of biopharmaceuticals and pharmaceuticals, or leveraging the YMC technology. Today's seminar is going to be between 15 to 25 minutes long, depending upon how many questions we get. We're going to leave plenty of time after the formal presentation to answer any questions you might have. This is one in a series of more than 10 recent webinars on chromatography for the application in the production of protein therapies. We invite you to explore these other webinars that cover a wide range of topics on the attributes of YMC's product platform. These are 15 to 30-minute presentations given by subject matter experts. Today's presenters draw from several of the YMC Centers of Excellence in the USA and Europe. Jeff Kakekli is the Applications and Marketing Manager at YMC America. Jeff's in charge of marketing, application development, and support, and technical customer support for all YMC liquid chromatography columns and media. Joining us from Germany is Anna Rusek. Anna is the Product Manager for Glass Columns in YMC Europe. She's responsible for project management, application, and technical support, as well as seminars. And Gerard Gatch, that's me. I'm the Chief Marketing Officer of YMC Process Technologies Biopharma Systems business, and I look after growth segments within the life science space and providing strategic direction for our systems business. This presentation will cover just a few of the products and services in the toolbox of pioneering and forward-looking purification products by YMC. Today, we'll briefly introduce you to some of YMC's chromatography columns, packing media, selection of LPLC and HPLC bench and GMP scale systems. Now, these are just a few of the lab and GMP process scale solutions for the biopharmaceutical and pharmaceutical industry that have been pioneered over the 40-year span of YMC Corporation. So I'll kick it off with a review of YMC from the 10,000 meter view, and then my colleagues will present more on the aforementioned technologies. For those of you who are new to YMC, YMC is a global technology supplier of purification technologies centered on chromatographic techniques and technologies serving both the pharmaceutical or the conventional small molecule and the biopharmaceutical or large molecule industries. Since 1980, YMC has been a leader in chromatography, producing bench and prep scale HPLC and LPLC systems, packing resins, columns, and method development. Additionally, YMC operates a CMO facility for the production of APIs and has labs in multiple of its facilities to assist customers in method development in Europe, India, USA, Korea, and Japan. YMC is a global company with manufacturing and offices in over 10 countries. An example, while many of the packing media and DAC columns are produced in or near the headquarters in Kyoto, Japan, the glass columns are produced in Germany, and the lab and GMP scale LPLC and HPLC systems are designed and manufactured in the USA and Japan. Okay, now I'll turn it over to my colleague, Jeff Kakekli, who will introduce us to the core products in resins and columns. Jeff?
Thank you, Gerard, for putting this webinar together and for inviting me to present some of YMC's preparative product offerings. I'll begin by highlighting YMC's core competencies, starting with production of spherical bonded phases. YMC produces over 30 different types of LC packing materials, ranging from 1.9 micron for UHPLC on up to 300 micron for large-scale prep purification. This includes traditional silica-based materials, modern hybrid silica materials, as well as polymer-based products. And these can be produced on a tons per year scale. Moving on to HPLC column production, we produce pre-packed columns from 75 micron ID for capillary HPLC on up to 300 millimeter ID custom prep columns. This includes stainless steel and glass columns, and Anna will cover glass columns in a few slides. And lastly, we also offer chromatography systems for prep and process scale, and Gerard will cover that after Anna's glass column section. On this slide, the main point I want to highlight is the dynamic axial compression columns. These are available in a variety of sizes from 50 millimeter to 1 meter ID and come in two main styles. The DAD series, where the piston compresses the bed from the top down, and the DAU series, which compresses from the bottom up. Both work similarly, but the DAD series makes unpacking a little easier. This also makes the unit taller than the DAU series, so ceiling height needs to be accounted for prior to purchase. Since I simply don't have the time to cover all stationary phases that we offer, I'm going to highlight the two main YMC packing materials that more or less go hand in hand with YPT and Chromacon's prep systems, with the first being YMC BioPro IEX. BioPro is a hydrophilic polymer-based ion exchange media that exhibits little to no secondary interactions, unlike traditional polystyrene divinyl benzene-based ion exchangers. It was specifically developed for biochromatography and is useful for IEX separations of monoclonal antibodies and other proteins, as well as peptides and oligos, to name just a few. It comes in two chemistries, sulfopropyl for strong cation exchange and quaternary amine for strong anion exchange. Both chemistries come in smaller, non-porous particles for fast, high-efficiency analytical separations, as well as a wide variety of porous particles in sizes up to 75 micron for prep scale. At process scale, BioPro IEX works extremely well for polishing after the protein A capture step. Next is YMC TriArt, YMC's flagship stationary phase. I'm going to cover most of these products in the next slide, so I won't spend much time here, but these are the main characteristics that make TriArt so attractive, especially the physical durability and scalability, which lends itself well to prep. I should also note here that we have two other phase chemistries that are not listed on this slide. We just released a new 200 angstrom version of TriArt Prep called Bio200C8, which is specifically designed for small protein and peptide purification, such as insulin. The other chemistry is a custom phase that we do not formally market, called TriArt Prep QA. This is a quaternary amine anion exchange material that we developed for a customer who needed a rugged base material that could handle harsh cleaning with organic solvent. I believe that memory serves me, it was either methylene chloride or chloroform. I like to mention this material specifically because it highlights the versatility of TriArt and YMC's ability to create custom products for our customers. So why choose TriArt? As you can see on the left side of this slide, traditional silica products offer the standard pH and temperature ranges. With its wider pH range, TriArt offers the ability to clean in place with acids or caustics such as sodium hydroxide with little to no ill effects. And while silica's mechanical strength has improved over the years, it still falls short of most hybrid products like TriArt. As I alluded to on the previous slide, TriArt's mechanical strength plays an important role for prep separations, with less fines created during unpacking and repacking, so you don't see the back pressure increases that you typically experience with silica. TriArt is also fully scalable. YMC actually manufactures the hybrid-based particle instead of purchasing it from a silica manufacturer, as is often done with silica-based materials. This means you can develop a method at the analytical scale and count on having predictable selectivity when scaling up to prep. So in the end, when you choose TriArt, you're choosing a stationary phase that has increased durability, greater versatility, and just better overall performance. And that pretty much ends my section of the talk, and I'll pass along to my colleague Anna for glass columns.
Thank you, Jeff, and hello to everybody from my side. Yes, YMC offers glass columns for low and medium pressure applications in laboratory and pilot scale. I'll give you now a short introduction into the 4 different column lines which are available. For laboratory scale applications, 2 different column lines are available. We have the Eco and Eco Plus line. Eco columns, which you see now on the slide, are designed for low and medium pressure applications. They are characterized by an easy piston adjustment due to the squeezed O-ring sealing principle. They are the perfect match for temperature-sensitive applications, as a heating cooling jacket is optional available. If the columns should be packed with SEC, resins, and longer bed lengths are required, 1 meter long glass bodies are available. The columns are available with IDs between 10 and 80 millimeter. You find the different IDs on the slide. And with regards to column lengths, 5 different standard lengths are available, also mentioned on the slide. This is the Eco Plus line. Eco Plus glass columns are designed for medium and high pressure applications. Of course, we are still in the field of glass columns for LPLC applications, so it's not the high pressure application like we know from analytical columns. But the 5mm ID columns have a pressure limit of 80 bar, and this is quite high for glass columns for liquid chromatography applications. The Eco Plus columns are characterized by an easy assembly with quick lock system. Due to higher pressure limits, you can increase the flow rate and generate this way faster runs. As the glass body is a calibrated precision glass, the Eco Plus columns give highly reproducible packing results. The columns are available with IDs between 5 and 50mm, again mentioned on the slide, and 3 different column lengths are available. Let's leave the laboratory scale applications and move on to the pilot scale columns. Like in laboratory scale, YMC offers 2 different column lines for pilot scale applications. In 2018, we've launched the brand new YMC Pilot Plus line, which you see here on this slide, and which is especially designed for pilot scale bio-LC applications. Next to that, we focused on easy and fast packing, operating, and unpacking. Due to the unique drain function, a bubble-free column packing is guaranteed. With the removable column body, unpacking is easy and clean. As the column is designed for bio-LC applications, the flow path is metal-free. This makes the columns 100% biocompatible. The columns are available in 4 inner diameters and 2 different lengths. Okay, let's move on to the second column line for pilot scale application, the YMC Pilot line. The YMC Pilot line is our flex line with a standard portfolio and the option of customization. Due to the optional heating cooling jacket, like shown in the picture, the columns are perfectly suited for temperature-sensitive applications. A solvent-resistant version with the end pieces made of stainless steel is available for applications where strong organic solvents are used. By customization, longer bed length ranges can be realized for SEC applications. The standard portfolio includes 4 inner diameters and 2 different lengths. Okay, let's summarize the key advantages of YMC glass columns. YMC offers high-quality glass columns for laboratory and pilot scale applications, which are designed and manufactured by YMC Europe and Germany. YMC glass columns have full compatibility with all common separation modes. An increased throughput is possible due to higher pressure limits. You will receive improved chromatographic results due to homogeneously packed column beds. And the maintenance for glass columns is fast and easy. They give an excellent column performance, and the glass columns show high cost efficiency. So now I will turn it back over to Gerard, who talk a bit about systems.
Thank you, Anna. Let me walk you through the systems technology provided by YMC. This isn't a comprehensive presentation on all systems, just a representation of several key platforms. The platform includes instruments for the Discovery or PD lab, and represented here, 3 of the YMC instruments. I'll spend a bit more time in the next slide on the Contichrom Cube unit shown here on the left, and that's the newest addition to the YMC system offering. These bench scale units scale to both pilot and production scale in HPLC and LPLC configurations. A bit more about these in coming slides. But now, as promised, I'll spend a little bit more time on the Contichrom Cube instrument. This is a novel tool that allows the user to perform routine HPLC and batch chromatography studies. But uniquely, it also has the capabilities to run twin column continuous modes for the capture step, the polishing step, and sequential chromatography, as well as a unique quality assurance tool called Enrich. We have webinars on each of these functionalities on our YouTube site, and I would invite you to explore, go deeper into this instrument, which has a wide range of conventional as well as next generation capabilities. Now for a brief review of the production scale systems. This shows examples of a batch or conventional single column LPLC and HPLC systems in the upper row. And these scale from anywhere from 0.1 to over 60 liters a minute with custom and standard designs offered. In the lower left quadrant are the newest addition to YMC. This is the patented Contichrom family, which is represented by the smaller cube instrument and its bigger brother, the GMP scaled up version called Contichrom TWIN, offered in either low pressure or HPLC versions with several different patented techniques embedded in each of those from the Contichrom family of patents. YMC is also a technology supplier, partner with Merck Millipore for buffered dilution skids, in which YMC have been an industry leader for precision dilution of concentrated buffers for the applications in biopharmaceutical plants at point of use or as a central dilution system. Now let me show you a little bit deeper look at some of the characteristics of just one of these units. In addition to using industry proven and mostly globally available components such as EcoDose pumps, Burkhardt valves, and E&H analytics, the systems are designed as you would expect, with options to enable UL Class 1 Div 2 hazardous location for certain applications or ATEX Zone 2, CE conformity. All our systems support 21 CFR Part 11 compliance, and most enable GAMP 5 compliance. While these are designed as standard platform, customers can request customization of certain components or configurations. Now, be assured that this series of systems can accurately scale from bench to production. This has been proven and documented by many of the top 10 global biopharmaceutical and pharmaceutical customers who we've interfaced with over the past 20 years. For more detail on each system, again, we have recorded several webinars that detail each of the systems, the hardware, the automation, some of the functionality, different modes of operation. So I invite you to, again, to explore deeper into each of these systems by reviewing these 15 to 30-minute long seminars by the subject matter experts. Now, we don't just stop there. We don't do just chromatography. Our division provides both standard and custom designs for things such as on an OEM basis, cell harvest systems, inline dilution systems, tangential flow filter systems. We do have simulated moving bed HPLC, blending, and buffer formulation systems. So this is just a sampling of some of the GMP downstream systems that are produced at YMC Process Technologies. So let me kind of bring it to a conclusion here with my colleagues. YMC has as you can see, and we only touched on a brief overview of the lab to full-scale production purification technologies, analytical columns. We didn't really get into the method development service. We talked about the GMP scale, pilot production scale, lab instruments, glass columns, and the preparative stationary phases. Okay, I'd like to bring back Jeff and Anna for the Q&A session. Really appreciate the questions that you have posed, audience. Continue to ask questions and we'll get to them. So let me review the first questions here. This one, I believe, is for Anna. What is the largest column that can be pre-packed, and what is a typical lead time?
So the largest one we can pack here is around 100 millimeter IB, so 10 centimeter. And the delivery time depends on the packing material. So we pack here YMC own resins, but also resins from other suppliers. So the delivery time depends then on the packing material and as well as specifications which need to be matched. So if there is a question on pre-packed columns, just drop us an email and we will check it.
Very good. And Anna, we're going to stick with you because looks like another question that would be in your field, and it is it possible to use your columns under GMP conditions?
Yes, of course. Our columns are implemented in GMP processes, so in laboratory and pilot scale. We can offer FDA, BCTs, E and USP Class 6 certificates. So for YMC Pilot Plus, for example, the brand new pilot column line for BioIC applications, the 3 types of certificates are part of the standard documentation. And in case of other certificates which are required, again, just yeah, drop us an email and we will check this for you.
Okay, thank you. It looks like there's a lot of interest in columns. Jeff, this one I believe you would be appropriate to answer. In reference to the DAC columns, why is the DAD model easier for unpacking than the DAU?
So with the DAD model, the piston moves in the downward position, whereas in the DAU it moves in the upward position. So the way you unpack these columns is by moving the piston, all the way to the end of the column, and essentially you have a column of used packing material that kind of comes either up and out of the top of it or down out of the bottom of it as the piston moves. So as it goes up, if you're using the DAU, it'll start going up, and you'll see this column of packing material coming out. You have to sit there with a bucket of some sort or a large beaker and a scraper and scrape it off into the beaker. Whereas with the DAD, piston's already coming in the downward direction, so really all you have to do is pop the end piece off, slide that bucket or beaker underneath there, and just use the piston to push that whole column right down out of the bottom, and it just falls right into your bucket. So that makes life a lot easier. But like I said during the presentation, you do have to take into account the ceiling heights, because the DAD tends to be a little bit taller, I think it's like a foot and a half taller than the DAU system.
Very good. This one looks like it would be for me. Question is, statement first, I am from Italy. Where can I see one of your lab systems? So we do have a lab that's been newly established in Milano. This is a lab that emanates from one of the founders of Chromacon. A professor, Massimo Mobidelli, has moved his lab from Zurich to Milan, and there would be fully equipped, and I'm sure that when appropriate, they would be able to invite people in to see systems there. But that's not the only place. There is both a office as well as 2 labs in Zurich, Switzerland, where there's a number of these systems that would be available for you to come in and take a look. I know that the team also has pilot rental for those systems if you need to have hands-on in your lab itself. Okay, here's one for Jeff. If you were developing a new method, analytical or prep, it appears you said hybrid silica products such as Triart would be a versatile choice. Is there ever an instance you can think where silica-based product would make more sense than a hybrid product?
So in the end, when developing a method, especially at the analytical, and usually it begins at the analytical scale, depending on your sample, how complex it is, how many different analytes you have or impurities you have, it's going to, if it's a very simple, pure sample, I don't think you need to worry about it too much, about screening a number of different columns. But if you have a lot of different compounds in there that you need, and you need to only purify, say, 1 or 2, it really comes down to selectivity. So I would use Triart as my first choice method development column. And I would see, being that it is the most durable and you can use it under the widest variety of conditions. But sometimes that selectivity is just not going to be right. So you may have to go back to a standard silica-based product, because selectivities can be different depending upon the nature of the base particle, the type of bonding used, whether it was monomeric or polymeric. Like I said, it just depends on how your compounds react chromatographically.
Very good. Thank you, Jeff. And Anna, this is a 2-part question. You mentioned bubble-free column packing with the YMC Pilot Plus columns. 1, how does this work? And 2, are there spare parts available?
Okay, yeah. The bubble-free column packing was one of the main aspects we wanted to address with our new pilot column line. So we have a unique drain function on the column. So it's a tubing on the right side of the column. And if you fill in the packing material and fill it up completely with the liquid, you can just insert the piston in a smooth angle and the excessive solvent will flow out. And this way, bubble-free column packing and piston insertion is guaranteed. So this is very simple and very easy. Spare parts are available for all YMC glass columns. You can just order an O-ring or a fridge, so just the spare part you need, and you do not have to order a complete spare part kit or something like that. So all spare parts are available.
Okay, fantastic. We're running, just looking at the time, we've got time for just a few more questions. So let me take one or two more here. And Jeff, I think it's over to you. Do you have any evidence or data that you could send us that highlights Tri-Art's durability? And before you answer that, can you also send that to me, so I'll make sure that I'll send anything out as a follow-up to this seminar?
Sure, Gerard, no problem. Yes, we definitely do have quite a few stability studies that show the durability of Tri-Art at high and low pH. Usually at low pH, you have cleavage of the stationary phase, which rears its ugly head as a loss of retention. And we have studies that show, at elevated temperatures and low pH that retention times remain relatively stable on out to, it might be like, I believe it's something like 200 hours. And we have the same thing at high pH. At high pH, you're literally, with traditional silica-based materials, you literally dissolve the silica base particle above like pH 7.5 or 8. And temperature only exacerbates that problem. So we do have studies that show at high pH, usually what you end up seeing is peak shape begins to suffer when the silica starts to dissolve. So we do have stability studies that show peak shape and plate count remain very good on out to 180 or 200 hours. But I can certainly send that to anyone who would like it.
Okay, thank you, Jeff. And I'll take the last one, and then we'll wrap up. This questioner says, what association do you have with Lewa? I remember seeing similar systems on their website. It's a good question. Formerly, there was a company called Lewa Process Technologies, and that company and its assets were purchased by YMC Corporation in 2018, the end of 2018, 2019. So it is essentially a continuation of the platforms that were developed under Lewa Process Technologies that has been assumed by YMC, and YMC also purchased a partner to that company called Chromacon AG. Chromacon was R&D and patents holder for many of the continuous chromatography systems that are now part of the YMC portfolio. Okay, so thank you very much, both Jeff and Anna. Really appreciate your presentations. Thank you to the audience for your participation today. Anyone who has an immediate application need, look on either of our websites or reach out to us at info@ymcpt.com. It's info@ymcpt.com. I will be sending you a follow-up link to an archived version of the recorded presentation and any reference documents that my colleagues forwarded to me supporting some of the products that they had reviewed today. Please share this with your colleagues who weren't available to attend today. And just a reminder, this is one in a series of webinars, all of which you can find on our YouTube channel on YMC. Just Google YouTube YMC Process Technologies, and you'll see the recorded versions of many different webinars. We'll end our session now. Thank you and have a good day.